Sorry, I got cut off on the last one. Just to remind you, these things can only be 15 minutes long, which isn't the worst thing in the world. It's just that sometimes I watch the clock better and make sure I have kind of a good breaking point, and other times not so good. But anyway, we were talking about this mitochondria because, just to review, you have hundreds of them or many of them inside each cell. I know when we saw her image of a cell, we just saw one. Some, like your muscle cells, are going to have far more than other cells. Your fat cells, not so much. And what they are somewhere along our genetic evolution, we had cells that engulfed them. It became a symbiotic relationship where the cells and the mitochondrial cell managed to live together for mutual benefit. They know this because the mitochondria has its own DNA. It has DNA that's separate from the DNA you get from your biological mother and father. And generally, when they do any kind of tracing somebody back to what their family heritage is, they're looking at the maternal mitochondrial DNA when they do that. But anyway, the mitochondria has an inner membrane and an outer membrane, and then a space in between. That becomes very important because these reactions that are taking place in there are highly combustible. So they are very unstable, high energy reactions that could not take place anywhere but in a mitochondria. Plus, there are also special chemicals that are found in the mitochondria, so enzymes that facilitate this whole process. We call the internal area the cristae, and that inner membrane, it's folded to increase surface area to allow for more and more proteins to be embedded to accommodate a multiple of these electron transport chain proteins to be embedded. And so where we are is we are at the electron. We're talking about the electron transfer chain. And what happened is we actually have an electron that's pulled off. Remember, we put that cofactor in, whether it's NAD or FAD, and we put a hydrogen atom on there. Well, we pulled that electron off, and now we are actually passing it down this electron chain. And each time it passes down the gradient, a little bit of energy is lost. And this has to happen in steps because otherwise it would be very combustible if it all happened at once. So it has to be a step-by-step reaction this way. But a couple of things that I wanted to say, because they become important to know. A couple of coenzymes strip those oxygen atoms off, and one of which is coenzyme Q, which you've probably heard of. Some people take supplements of coenzyme Q. That's one of the enzymes that is going to strip FAD and strip the hydrogen off that to release that hydrogen ion to pass down the electron transport chain. The thing that picks it up are these cytochromes. These proteins that are embedded in the inner membrane are called cytochromes that have the capacity to actually hold that electron. And the cytochromes are cytochrome A, B, and C. And the hydrogen cyanide gas, which you've probably heard of, what it does is it actually attaches to one of these embedded proteins, these cytochromes, and that's why it's potentially harmful. Because of that, it basically reduces your ability to make ATP. Dramatically reduces it, because basically stops the whole system. But anyway, the electrons are pulled off, and inside the inner membrane of the mitochondria, sent down this gradient is sent down this electron transport chain by these cytochromes, and it has to be done in steps because it's too reactive to be done all at once. So that's why you have this picture, this oxidative phosphorylation, and that's the term that your book tends to use, that's going on in this electron transport chain. Each time that electron bounces down a step, energy is given off. And then using the energy gained, so down that steps, in that what we're calling now the hot potato toss, those cytochromes pump hydrogen ions, because what we did was we stripped an electron off that hydrogen. So now you have in the nucleus a proton and a neutron, so you have a hydrogen ion with a positive charge into the inner membrane. So it gets pumped in because it's not lipid soluble. It can't actually diffuse. And then the high numbers of protons put into the inner membrane space becomes a potential energy. Going back to again 141, where you have these concentration gradients and electrical gradients where they want to stay in equilibrium. Well, if you're pumping out a tremendous amount of something that's positively charged, then you have a deficit inside, so a negative deficit, so those positively charged ones want to come back in. And that actually is what happens. So you have a concentration gradient with regard to the hydrogen ions plus an electrical gradient. And what they end up doing is then coming back in through channels. So this is all happening in the mitochondria. So they are pumped from the inner portion of the mitochondria to the outer portion still in the mitochondria. That concentration electrical gradient sets up, so now they're actually going to move back in. The channels move through protein channels with the help of an enzyme called ATP synase, so that it actually is embedded in that channel, embedded in the inner mitochondrial membrane, and they're tied together with this synthesizing this enzyme that can synthesize ATP, and that's where the phosphate is put onto ADP. So this is where the actual ATP is created, not so much where that electron transport chain, where those electrons give off energy. That energy then is used to run this pump that pumps the hydrogen ions out of the inner membrane, but then when they come back in, that also creates energy, which creates ATP. And then the final event, the three cytochromes pass the electron to 1/2 of a molecule of O2. That O2 becomes negatively charged and picks up 2 hydrogens because these hydrogen ions now have a low enough energy level that they won't combust, and that then forms H2O, otherwise known as metabolic water. And that in that oxygen becomes the final electron acceptor, and that becomes an important thing to know too. That's where we make water in this particular process, and we make water because oxygen then can be the one that gets rid of, or not just takes the final product, which is again outside of the mitochondria. This is potentially dangerous to have this hydrogen ion, but water takes it. Oxygen takes it, turns it into water, which is something that the body recognizes and can do something with, can actually push it out of the cell, and it can mingle with the interstitial fluids. Other players in cellular respiration, just because we are talking about nutrition, B5 is needed to form coenzyme A. Riboflavin and niacin 2 and 3 are used to make those cofactors. So your B vitamins, when they talk about take B vitamins for energy, now you can explain a little bit about why that's the truth. Oxygen or carbon dioxide is produced and given off, and then the metabolic water we just talked about, that's the H2O that is formed as the end result of that electron transport chain. And then a little bit of a summary if you need it all together. The glycolysis happens outside the mitochondria, so in the cytoplasm. The Krebs cycle, TCA cycle, citric acid cycle takes place within the mitochondrial matrix. We call that a matrix because you have the inner outer. And the cytochrome proteins are embedded in the inner membrane. And that protein pump is in the inner membrane, moving those protein positively charged ions back and forth to create energy, to make ATP. If glucose isn't needed, so moving on, if glucose isn't needed, it's going to be stored. And the stored form of glucose is, or the process of making stored carbohydrates is called glycogenesis. And that can be stored in your skeletal muscles and your liver cells. So you can store some, not a whole lot, but the process is called glycogenesis. So building up into polysaccharides. And then when you have a time of need, so remember what you're doing all this for is to have a certain blood sugar concentration, so blood glucose level, because that's what your brain likes. Your brain likes a certain blood glucose level. And so when you don't have readily available glucose, then the stored form can be broken down, and that's called glycogenolysis. Is the opposite of glycogenesis, and that's when the body activities require ATP. So glycogen is broken down and released into the bloodstream so it can be taken to the cells and used in this whole cellular respiration process. So basically you're understanding what these terms mean here. And then glyconeogenesis, sorry, I didn't make these words up so that they're all similar, is the ability to form glucose from its metabolites, or its metabolites from fat or protein. So to use fat or protein in this Krebs cycle, or the citric acid cycle. Particularly in fasting, starving, or if you happen to be consuming a low carbohydrate diet. So the ability to use something other than carbohydrates to fuel this same chain. In terms of the Krebs cycle, we will actually we're kind of going to talk about how other metabolites are broken down. Amino acids, glycerol portion of triglycerides are used to make glucose. All we need used to form glucose molecules are pyruvic acid. So all we need are the 3-carbon chain, because that's the definition of what pyruvic acid is. So somehow or another we need to get a 3-carbon chain off those other metabolites. Moving on to those lipids. We are going to talk about carbohydrates, lipids, and proteins in this particular chapter. Lipids are waxes, steroidal hormones, fat-soluble vitamins. So there's a component to fat-soluble vitamins. Monoglycerides, diglycerides, phospholipids. Remember phospholipids of your cell membrane. Triglycerides are what we're going to talk about. It's a condensed form of lipids, in its condensed form it's for usable energy. So triglycerides are what we commonly call fat. And here's a triglyceride molecule, saturated and unsaturated. The glycerol is the backbone, so you have glycerol here in red, and the backbone with 3 fatty acids. So actually, unfortunately, this is just one, and we need to move on to the next one. So we'll pick this up on the next one.